I'll introduce myself. My name is George Loizou. I'm from the UK Health and Safety Laboratory, which is a research laboratory of the UK Health and Safety Executive, which is a government agency charged with protecting the health and safety of working people. I'm going to talk about physiologically-based pharmacokinetic modeling because it's a tool that it's becoming increasingly important in biologically based chemical risk assessment and nanoparticle assessment. The outline of the talk, I'm going to describe what a PBPK model is. I'm going to talk about how it's used as a research tool, because it's actually fundamentally important to understand thinking behind modeling. I'm going to talk about how it's applied in the risk assessment approach. I'm going to give you an example of how it's applied. And you'll probably notice that there's the chemical styrene up there. I have to give you an example based on chemicals because that's my experience. Originally I was asked to talk about nanoparticles, but I actually don't model nanoparticles. But the fundamentals of PBPK are the same, whether it's for chemicals or nanoparticles. So I'll give you an example a real example based on styrene. I'm going to briefly talk about why this technology, up until now, hasn't really been taken up quite to the extent that one would expect because of the power of the technology. And this is primarily born out of the experience of working within a regulatory agency where you have to deal with regulators who, by and large, are biological scientists who are not very numerate and have interesting responses to mathematical equations. And how we have tried to address these issues. An apology, there are some vestiges on my slides of a presentation I initially was expecting to present, which was to end in a hands-on demonstration of a PBPK model from a chemical point of view, but we removed that and that's going to be replaced by Lang's demonstration. So there's one or two references in my slides and one or two slides that are actually not directly relevant to what I'm going to talk about today. So what is a physiologically based pharmacokinetic model? Well, this shameless animation is always used to try and impress people, to show that we're actually trying to describe real dynamic biological processes, but clearly we have to simplify them and build up and add complexity as and when required. So it's a biologically realistic yet simplified emulation of a real organism that you wish to study. So it's a mathematical description of a biological system which we translate into computer code, and we can then solve these equations because of the onset of computing power. Now, I had a chat with various people at lunchtime, so I apologize to those who know more about mathematical modeling and computing power than I assumed in the previous group, because I talked about how this whole technology really took off in the mid-80s when desktop computing power increasingly became available. So if you look at the sort of publication rate of PBPK modeling, it seems to correlate with CPU availability in desktop computers. So it's an independent structural model. Basically, we describe all the organs and tissues in the body that we believe are important in understanding where and how fast chemicals and particles distribute around the body. And they're all connected correctly by the vascular system. And we have to generate chemical-specific information, which we superimpose over model predictions. We go to great lengths to explain that we don't actually tweak parameters and fit lines through data. The first and best way of approaching this type of modeling is what we call a priori modeling, where you capture your knowledge and exercise the model and look at the output of the model compared to real data. This is how PBPK models are represented in the literature. You have these boxes where you have various organs represented. The liver is invariably represented in a PBPK model because the liver is the chemical factory of the body. It has by far more metabolic capacity than any other organ and tissue. So as we want to start with the simplest model first, we can assume that most of the quantitative contribution to the amount of metabolism that goes on will occur in the liver. And only if we know something about the substance that we're studying, perhaps it's toxic to the central nervous system, then we'll describe the brain discretely and perhaps ascribe metabolism to the brain because it's a target organ. There are two compartments there, the slowly and richly perfused. This is ceasing to work now.
Yes, ma'am.
I'll use that. Okay, thank you. So there's the slowly and rapidly perfused compartments, and we usually start with those, and these are aggregated compartments. The rapidly perfused compartment contains all the viscera, the gastrointestinal tract, the spleen, the pancreas, the kidney. These are all rapidly perfused in terms of their blood flow. The slowly perfused tissues are bone and cartilage, muscle, fat. And when we expand the model and make it more complex, we take the organs or tissues we're interested in out of those compartments and expand the model. And we do this to ensure that we never build a model that exceeds body weight. So you can imagine a situation where you might add compartments and body masses and inadvertently find that they add up to more than body weight. This is just to emphasize the biological realism of our modeling, where we have images of organs and tissues in the compartments. Now, in occupational toxicology, environmental toxicology, we are overwhelmingly interested in the inhalation and dermal routes of exposure. If we were in the pharmaceutical arena, we would have a gastrointestinal tract compartment defined discretely with a substantial amount of detail, because in the pharmaceutical arena they're interested in oral administration of drugs and the uptake of drugs along the entire length of the gastrointestinal tract. So what do we need to populate our model? We need anatomical and physiological parameters, which are readily available in the literature, have been around for a long time. So organ and tissue masses and blood flow rates, regional blood flow rates through these organs and tissues. Biochemical parameters, things like metabolic rate constants for metabolism, binding to various receptors, and physicochemical parameters. These are measures of solubility of chemical in biological media. We call these partition coefficients. And here's a sort of, I'm positing a difference between chemicals and nanoparticles. I understand that it's not possible to generate partition coefficients for nanoparticles, so you have to find another way of describing the movement of a nanoparticle from the blood into an organ or tissue. And I understand you would use an empirical rate constant for that kind of thing. And again, this is a slide I put in to try and expand on that. So if a nanoparticle is in the body, it can penetrate organs and tissues by movement through an intercellular cleft. It's a gap between the cells of the endothelium of the blood vessels. It can, if it's lipophilic, it can perfuse directly through the lipophilic membrane, or it might be introduced into the organs or tissues by a process of pinocytosis. So generally we don't have to take that kind of mechanistic information into account with chemicals. Let's just reiterating what I've just explained. I'll give you an idea of the type of data. What does it look like First of all, the bell next to the 15 should be a multiplication sign. I think this happens when we use an older version of PowerPoint with a newer version. Certain symbols get jumbled up and incorrect. So imagine we're trying to simulate a human being who's 75 kilograms in weight. We can estimate their liver mass by assuming that about 2.7% of body weight is liver. So they will have a 2 kilogram liver. And under sedentary conditions, which is as you are now, sitting without doing much work, apart from intellectual, you have about 1.5 liters of blood perfusing the liver per minute. So about twice the amount of volume as this bottle is flowing through the liver every minute as we speak. Conversely, imagine or assume this individual's fat is about 19% of their body weight. They may have about 14 kilograms of fat. And this is all the different types of fat. We're sort of simplifying the body and we're lumping it all together as one compartment. And you get about 300 mils of blood perfusing the entire 14 kilograms of fat. So about half the amount of volume in this bottle is blood perfusing the fat compartment. So there you have a good demonstration of richly and slowly perfused tissues. And you can see the other kidney and the brain are rapidly perfused tissues. And the muscle is quite a slowly perfused tissue. And we can estimate alveolar ventilation rate and cardiac output using some pretty good allometric equations. 15 times body weight to the 0.74 gives a pretty good estimation of how much you inhale and exhale. About 6 liters a minute under sedentary conditions. So that's the type of data we put into our models. The research approach. There's a more well-known conventional form of pharmacokinetics, which is data-based, it's empirical, it's not physiologically based. And under those circumstances, which is a very useful tool within the pharmaceutical arena when you want to interpolate between different people calculating doses within the same human species, it's rather useful in that you have your assumptions, you design your experiment, you get your data, and then you fit lines through the data by tweaking parameters. And you get parameters like half-life and volume of distribution, and these are useful. They tell you how long a substance takes to pass through the system. There are more abstract parameters like volume of distribution, which is an indication, one way of thinking of it, an indication of how soluble the chemical is. If the substance is very lipophilic, soluble in fat, it will have a bizarre volume of distribution which may be 200-300 liters. This is just an indication that you need that much of an aqueous phase to dissolve a small amount of this substance, but it's an abstract parameter. So if you're happy and you fit lines through the data, you get that kind of information, you're happy and the story ends there. With the theoretical approach, the physiologically based approach, what we do there is we think of the problem first, we try and capture what knowledge we have, biological knowledge, we translate that into a model, we exercise the model and look at the output of the model, and only then do we compare it to data. And in the first instance, we're interested in the qualitative appearance of the data. What's the shape of the data and what's the shape of the output? Are they close? If they are close, we're reasonably happy. Generally speaking, we talk about being within 2 standard deviations of the data, because that kind of difference can be explained by inter-individual variability. I'm going to give you an example of the approach, the research approach, which was also required as part of the applied approach. 15 years ago, when I first went to the health and safety laboratory, one of the first projects I had was to look at a problem that the health and safety executive were dealing with, which at that time they were still a regulatory agency. They're no longer because of REACH. But they were regulating a number of industrial solvents like toluene, xylene, ethyl, benzene. People were using these substances in various workshops, being exposed to these vapors, getting drowsy because it has a subnarcotic, they have a subnarcotic property. And so the health and safety executive wanted to set standards where obviously you will not have people that are drowsy in the workplace who are using equipment that might be quite dangerous. But on top of that, they realized that, as is quite common, particularly in the UK, people go out to the pub at lunchtime and have a couple of beers, especially people that work in garages and workshops. And it turns out that the enzyme that metabolizes all these solvents also metabolizes ethanol. So if you have a substantial amount of ethanol present in your body, it's going to compete for metabolism with the solvent. And by and large, that means that more solvent will remain in the bloodstream and be available to be taken up into the brain and cause an increased drowsiness. So we had to review their occupational exposure standards, taking on board the presence of ethanol. This was one of the first exercises they wanted us to look at to demonstrate to them how useful PBPK modeling might be in the future for regulation. So we built a simple model for ethanol, and we found some measured data in the literature going way back as far as 1977, where a bunch of Caucasian, white male Caucasians, which was and still is predominantly the species that is used in human volunteer studies, and they were given ethanol diluted in fruit juice, various concentrations of ethanol, ranging from about 4 to 45 grams diluted in fruit juice, and they were asked to imbibe this stuff as quickly as possible. And the symbols are measured concentrations, venous blood concentrations of ethanol. The solid lines are our model predictions of that experiment. And as a toxicologist, biochemist, I was very impressed with this, very happy with this. But my more rigorous mathematician colleagues were more interested in why there appears to be a delay in reaching the peak concentration at the highest exposure concentration. So if you look at the red symbols and the blue symbols, or the purple ones, and compared to the simulations, there seems to be a delay. And that sent me thinking, and that sent me back to the literature, and it said there's a deficit in our knowledge. We need to improve the model to explain that observation. And indeed, it turns out that not only ethanol, but various other drugs, paracetamol for example, cause an inhibition of gastric motility. So there's the normal peristaltic function of the gastrointestinal tract, which is the contraction of muscles which propels food and drink along your gastrointestinal tract as your body extracts nutrients from your gut. And ethanol numbs, inhibits that process. And we surmised, we hypothesized that it was a concentration-dependent inhibition of this gastric motility. So I described the biology to the mathematicians, and they came up with a simplified description of the uptake of substance from the gut. The broken blue line signifies the introduction of a bolus dose of something. A bolus means a large, sudden introduction of a drug or a chemical into the body, in this case the stomach. And the turquoise broken line signifies an effect on the muscles of the stomach that cause contraction. And the same thing's happening in the lower GI tract. And together these processes modulate the amount of substance that appears in the portal blood that feeds into the liver. And together this should help us describe a concentration-dependent inhibition of metabolism. And we added that model to the original model, ran the model again, and got an improved fit consistent with our hypothesis. And this basically is the way that we move forward with PBPK modeling. It captures our knowledge, it allows us to do hypothesis testing in a research scenario. This slide serves a number of purposes. I'll go back to explaining that conventional pharmacokinetics will allow you just to fit a line through one part of the data, probably around here, and you would, from that part of the data, get a half-life. And you will say xylene in the body takes 12 hours to flush all the way through. This is a study conducted at the Health and Safety Laboratory. We were able, particularly in the past, to do some human volunteer studies, and they were interested back then, this was 20 years ago, in extended work shifts, people that were working for more than 8 hours a day. And here they exposed people to 40 parts per million xylene, and they were also testing a breath sampler, because another aspect of the work of the Health and Safety Laboratory was finding ways, non-invasive ways of estimating body burden. You can't take blood from workers. They, by and large, won't allow that, and it's not ethical, it's not important enough. So we have to find non-invasive ways, either urine sampling, saliva, hair, and breath. And in this case, they were testing this breath sampler. They were exposed in the exposure chamber. They were allowed to step out of the chamber for half an hour, and these are these two troughs here, and then go back into the chamber. And then this is a 12-hour overnight break before they go back into the chamber for 2 exposures. The symbols are measured exhaled breath concentrations. What I'm going to show you in the next slide is this same simulation, and you'll be able to see that this single line is actually composed of a whole number of other lines, contributions from other organs and tissues, which you can separate out when you use PBPK modeling. I'm going to apologize from now because, as I said before, there's an earlier version of PowerPoint that I used, and unfortunately a black bar appears over this in the wrong place. But when you see the animation, the top bar that moves up and down corresponds to the brain, and then the kidney, the muscle, the fat, and the liver. The slide on the previous graph will be recreated in the bottom panel, and the line will be broken down into the components that occur in the liver, the muscle, the fat, the brain, and the kidney. So as exposure is progressing, you can see the profile in the previous slide reproduced at the bottom. You can see the red bar, that corresponds to the fat compartment. You can see the richly and slowly perfused tissues, liver, kidney, brain. They fill up rapidly and they empty rapidly. But the slowly perfused tissues fill up slowly. But the fat compartment sequesters the substance because xylene is a lipophilic chemical. So it will retain in the body. And you can go over 2 days and find that you haven't flushed out the substance from the previous day's exposure because it's sequestered in the fat compartment. And this can go on 5 days a week. Maybe a weekend isn't enough time to flush your body out. This kind of information is being used in America to send people to health farms, people that work on airport runways that get exposed to jet fuel vapors, and they say, all right, send them off to these health farms for a month so that their fat compartment will empty from all these solvents, and then bring them back. But the important thing about this is this information is extremely useful for biological monitoring. This will help you decide when to take a sample and how to interpret that sample. What is it telling you? What's the information that's captured in this biological monitoring sample? So what are the strengths of PBPK modeling? Because the models are mechanistic, the mechanistic nature allows us to explain data, not just describe data. Importantly, we can actually estimate how much substance is delivered to the organs and tissues, and this is what we call tissue dosimetry. And we can correlate the concentrations in the cells with a response. And this is a far superior way of trying to correlate exposure with disease incidence than trying to correlate concentrations in the atmosphere with response, which is the current epidemiological paradigm. So this tissue dosimetry is the linchpin of this data-informed, biologically based risk assessment. So, as I've just said, it's a tissue dosimetry is a better way of correlating disease with response because it's the biologically effective concentration that we're actually estimating. And we try and capture as much information about what's going on in the cell, not only the amount that gets delivered to the cell, but what is the form of the chemical in the cell. Is it the parent chemical you were exposed to? Or is it a metabolite or metabolites? And where do those metabolites go? Are they binding directly to DNA or to some other macro molecule in the cell? And what is the nature? Is it area under the curve that you're interested in? Is it the peak? There are a whole load of dose metrics that we can capture using knowledge from all over the place in the model and allow us to then exercise the model and try and correlate that with a response. So, now how PBPK modeling is being used and increasingly being used in this more biologically based risk assessment paradigm. The environmental toxicology, industrial toxicology arena is a human data poor environment. The data I just showed you is very rare, and it's obviously not with a toxic substance. We cannot do experiments on human beings. So we're reliant on extrapolating from animals and increasingly from alternatives to animals to humans. And PBPK is very, very powerful for this exercise. You can think of it logically. If you have a model for the rat with all the organs and tissues, you can just change the masses and blood flows to those of the human, and you have a model for the human. The difference is that we can ascribe passive absorption and distribution processes are the same across species. Logically, a substance that is soluble in the fat compartment is very likely to have very similar solubility in rat fat as it does with human fat. The differences come when you look at active processes, enzyme activity, proteins and transporters in the gut that require energy. Those are where we see differences between species. And if we can measure them, we can incorporate them into our model, and we can accommodate and do a very good interspecies extrapolation. So the approach to risk assessment, which is common to all risk assessments, irrespective of the use of PBPK, is to search for the critical study, a toxicological study in which you have identified the most sensitive species and the most sensitive measurement in that species that correlates with a toxic effect. And you try and identify the appropriate dose metric. As I said before, what is the appropriate form of the substance in that study that you believe is causing the toxic effect? This graph, this slide just wants to emphasize the point that as you go from exposure concentrations in the atmosphere to concentrations down at the cell, you get a far better correlation with response. So the example is with styrene. Styrene is a high volume chemical. Chemical industry has made huge amounts of money from this substance. But every so often people come up with studies that indicate that it's actually carcinogenic. And they often find that the study has been conducted in a particular strain of mouse, in this case the CD1 mouse, where they've observed tumours in the bronchoalveolar region of the lung. And that requires investigation. And so, because of the importance of the chemicals, the industry is prepared to invest and generate more data to try and keep this very valuable and important chemical on the market. So, they looked at the database and found tumours in the lung of that particular mouse, but they didn't find very much evidence of toxicity in humans and not much in other species. So the most critical and sensitive endpoint is bronchoalveolar tumours in the CD1 mouse. A little bit of chemistry. So styrene is bioactivated, metabolized to styrene oxide, which we are sure is the genotoxic carcinogen, the substance that binds to DNA that eventually leads to tumors. There's another enzyme called microsomal epoxide hydrolase, which detoxifies styrene oxide. So you have bioactivation to a toxic entity and removal of that toxic entity by microsomal epoxide hydrolase. And this is the region where these tumors are observed, in the bronchoalveolar region of the lung. Some interesting information. In the mouse, about 80% of the clara cells are the cells with the highest metabolic capacity compared to any other lung cell, occur in the bronchoalveolar region of the mouse lung. Similarly, in human beings, 85% occur in the bronchoalveolar region, but only 42% in the rat. What else do we know? We know that the mouse has the highest activity of the cytochrome P450 enzyme, which activates styrene to the toxic entity, and the lowest activity of microsomal epoxide hydrolase, which detoxifies styrene oxide. And it's the opposite in humans. Humans have the lowest P450 activity in the lung and the highest microsomal epoxide hydrolase. So you would say that the humans are best able to deal with styrene exposure. And that perhaps explains why we don't see any epidemiological evidence for lung tumors in human beings. And this just is an illustrative slide showing that if you try and plot concentration of styrene in inhaled concentration in the venous blood, you don't get a great correlation. But if you try and plot the tissue concentration, you start to get a qualitative agreement. The shape of the data and the shape of the model predictions are similar. So the last couple of slides. Mode of action refers to those important obligatory biochemical reactions that must occur so that before you observe toxicity. That's what we need to capture in our model. We do not need to capture every single detail, which is the mechanism of action. Thankfully, that is not necessary. Obviously, because mechanism of action is rarely available in total. And this mode of action, these are just a few slides of interest, generally speaking, as I finish now. Mode of action is the basis of an international program of trying to harmonize risk assessment globally. And increasingly, we're observing that there are a number of processes that precede apical toxicity. What I mean by apical toxicity, this is overt, full-blown pathology. There are a number of processes, less than 10 at this moment in time, that we can measure that precede all of these diseases. And perhaps we can develop in vitro systems where we can measure these processes, just looking at perturbations of biochemical pathways. This will allow us to do a risk assessment for a whole range of disease endpoints in vitro. But of course, these have to be linked with dose, duration, and response, and so that's where PBPK modeling will come in. And there I've finished, and this is a slide which is of no relevance to the conversation now because it was assuming I was going to do a hands-on demonstration. So if you have any questions, I'm happy to answer them. Thank you very much for your attention. It was all crystal clear. Oh no, there's one.
Kinetics, so you also predict how the concentration decreases with time. But what kind of calculation does it involve?
I think you're referring to this morning's exercise where you have these input. Now, with PBPK models, all you have is an exposure concentration So it could be an inhalation exposure concentration, and that's all you input. And then the rest is calculated. The model will calculate the amount that's delivered into the blood using this inhalation rate, the partition coefficient, how soluble the chemical is, which is part of the mass balance equations, which is used to calculate how much gets into the blood. And then once it's in the blood, the cardiac output and blood flow rates that distribute the chemical around the body, that's calculated all for you. So it's only inhalation is the input, apart from all the other.
This much I understood. And then how do you calculate how much of the substance remains in the tissue?
Well, you have differential equations that calculate the rate of change of substance in all the organs and tissues and compartments that you've described.
Do you need all this rate coefficients too?
Yes, yes. In another presentation, I would have given you the problems and criticisms of PBPK modeling and why it hasn't been taken up. And that is one. It's data intensive, complex, perceived as complex, resource intensive, time consuming, and you can only use one chemical at a time. And our research over the years has been to address those issues to the regulators, which we think we're doing quite well with tools, user-friendly tools that allow a non-numerate toxicologist to build models without having to worry too much about the maths. So they see a biological user-friendly front end, they answer biological questions, the maths is generated underneath for them, converted to computer code. You can select the code that you wish. You can export in MATLAB code, Axel, Berkeley Madonna, or MCSim. These are 4 simulation software packages. We're wanting to include R, the freely available statistical modeling package, so that you can have a totally free-to-use system from start to finish. And it's some of the courses that I run, I actually use this slide to explain that we actually build a PBPK model from a peer-reviewed paper, and within 10 minutes we've built the model and we've recreated a figure from that paper. Usually building a model, I'm sure you know, can take over a day, maybe a few days by the time you've built it, debugged it, and all of that. We can do quite a few of the published PBPK models to date. We can build them in 10 minutes.
So for the PBPK model, basically you have some diffusion constants, and partly they are defined by the bloodstream, right? So is it very different between different types of animals, or let's say animals versus humans, for the different organs, for example?
The rates. Yes. You have a mouse and a human, and there's a thing called pharmacologic time and chronologic time. If you divide respiration movements and heartbeat, it's around 4. There's a constant ratio. So there is a logic. Basically, a mouse lives its life at a far higher rate than a human being. So a human being has like 70 beats a minute. The mouse, I think, 200 beats a minute. The elephant, 30 beats a minute. If you take all these things into account, you can normalize from across species. Is that?
Yes, and just a second question. And are they, well, because I'm not in the toxicology field, are these chemicals, do they have a different impact in the same organ for different species, assuming we have already these constants about the
Yes. Yes.
It's
the example I gave you is an example. You had tumors in the mouse, but we have no evidence of tumors in humans, and it's because of the biochemical differences. And this is the argument that the companies put to the regulators. They say, look, it causes tumors in the mouse, but it's not likely to, and there's no evidence of it causing tumors in humans because the enzymology is different.
Sorry, now the next question. Because I can understand more or less fairly how this model works with simple organic molecules, but what's your perception about the future, the possibilities of the model with particles?
I'll respond first in a general sense. If you understand, if you have a hypothesis in your mind about how something works, you can model it. Just, if you're not a mathematician, you'll talk to a mathematician and he'll model it for you. So that's the first thing. Modeling is a platform for hypothesis testing.
Yeah, but there is a very important difference between particles and chemical substances. That is that in a compartment with a chemical substance, you will have an homogeneous concentration, but this is not true with the particles. So, do you think these kind of models can be fit?
Yes. Why do you think, what are the differences? This not homogeneous, you have already described the difference. If you analyze those differences, if you talk to a mathematician, they will hopefully come up with mathematical explanation of the differences, and you'll put that into your model. It's that simple. I mean, there's some mathematicians here. There's a physicist. I'm sure they can...
Yeah, but I mean, we can study very easily, I don't know, chemical kinetics, the rate constants and everything for a homogeneous substance, and we still don't fully understand the behavior of a particle in water. So that's why I'm asking this question. Maybe it's a very difficult question to answer.
No, the answer is we have attempted to do that. I think the mathematics are the same, and the principle by and large are shared between what originally been done in PBPK with chemicals. So there are lots of things that the modeling of particle learn from PBPK. The scaling that George talked about, the physiological time versus the chronological time. You need to do that even for scaling from human from rat to human for particles. So there is that element in there. For particles,
speak up a little bit.
for particles, at the moment, as George pointed out, the limitation with particles is simply because we are new and the database is less than what the chemical.
No, I understand this.
So we are making our way there, and I hope to show also is that with particles, the traditional problem has always been at the portal entry. When George talked about chemicals, it shows that it's distributed all over the body. That is new with nanoparticles. And even with nanoparticles, 99% of it are still retained at the portal entry. Some of it actually go beyond into the blood and so on, but the number are small. So the idea of something beyond the portal entry is something new. And we are making progress is to understand how particles is biodistributed in the body. For example, we know that it's size dependent. That if you inject nanoparticle in the blood, the larger nanoparticle can accumulate in the lung, but the smaller nanoparticle goes straight into the vision. So there's a difference in phenotype that is a function of size. There's also the difference, of course, size also being more translocation than other. So big particles, for example, that you deliver through the lung, stay in the lung. But the smaller they become, the more likely they can be diffused beyond the lung into the blood.
Lang, you're going to give that presentation when I've...
One last question, very brief, because, well, there is a chance that you can convince me about this, and it's related with some particular kind of nanoparticle that is not engineered nanoparticle, and maybe you are more familiar with it. The viruses, because the viruses are somehow nanoparticles, they are in the nanoscale. you know of any application of these kind of models for estimating the time of residence of a virus in a body?
Yeah, yeah. I don't understand why you think it's a problem. I really don't. If you know nothing about something, you can't model it. The value of these things is it's a platform for hypothesis testing. You capture your knowledge, and the reason why we need to do it is because I personally, in my mind, cannot calculate the blood flow through the liver, the solubility of the chemical in the blood, metabolism once it gets in. I can't do all that in my head, but the computer can do that.
Yes, yes. But well, anyway, it doesn't matter. But about the virus, do you know of any application?
I vaguely know, and yes, I mean endotoxins are products produced from bacterial infections, and they try and sort of model bacterial infection, proliferation, production of endotoxin, viruses, because some of those are thought to cause cancer. You know, they get in, and yes, you can model anything, absolutely anything.
Of how bacteria enter the lung.
I think we'll move on with Lance lecture.